In our previous part 1 video, we had discussed about the DNA replication introduction in detail. Then we had studied about the three models for DNA replication that we had discussed in detail, that are semi-conservative DNA replication, conservative DNA replication, and dispersive DNA replication. And lastly, we had discussed about the semi-discontinuous DNA replication. Now in part 2 video, we will discuss in detail about the prokaryotic DNA replication, eukaryotic DNA replication, and lastly the significance of DNA replication. Now first, the prokaryotic DNA replication. The basic features of prokaryotic DNA replication is that replication is bidirectional, which means it is a type of DNA replication where replication is moving along in both directions from the starting point. This creates two replication forks moving in opposite direction at a single origin of replication. Prokaryotic DNA replication takes place in the cell cytoplasm. Prokaryotic DNA replication synthesis occurs only in the 5' to 3' directions. Individual strands of DNA are manufactured in different directions, producing a leading and a lagging strand. Lagging strands are created by the production of small DNA fragments called Okazaki fragments, and Okazaki fragments are the short sequence of DNA nucleotide which are approximately 150 to 200 base pair long in eukaryotes, which are synthesized discontinuously and later linked together by the enzyme DNA ligase to create the lagging strand during replication that are eventually joined together. Now, let's talk about the enzymes for prokaryotic DNA replication. These are helicases, single strand binding protein, primase, DNAP third, which is abbreviated as DNA polymerase third, DNAP1, which is abbreviated as DNA polymerase 1 enzyme, then DNA topoisomerase 1, DNA topoisomerase 2, and lastly DNA ligase. Now let's discuss about in detail their functions of the enzyme. First, helicases. They unwind the DNA helix at the start of the replication. Next, single strand binding protein. Their function is to bind to the single strand of unwound DNA to prevent the reformation of the DNA helix during replication. Next enzyme is primase. Their function is to synthesize the RNA primer needed for the initiation of DNA chain synthesis. Next enzyme is DNA polymerase III. It elongates DNA strand by adding deoxyribonucleotide to the 3' end of the chain. Synthesis can only occur in the 5' to 3' direction because of DNA polymerase enzyme third. Now, DNA polymerase enzyme first. Their function is to replace RNA primer with the appropriate deoxynucleotides. Next enzyme is DNA topoisomerase 1. Their function is that it relaxes the DNA helix during replication through creation of a nick in one of the DNA strand. Next enzyme is DNA topoisomerase 2. It relieves the strain on the DNA helix during replication by forming in the helix through the creation of nick in both strands of DNA. And the last enzyme for prokaryotic DNA replication is that DNA ligase. It forms a 3' to 5' phosphodiester bond between adjacent fragment of DNA. Now, the steps of DNA replication in prokaryotes. Here in this figure, what is shown that DNA replication begins at a sequence-specific spot on the DNA molecule called origin of replication. At the origin, enzyme unwind the double helix, making a component accessible for the replication. The unwound helix is stabilized by single-strand binding protein and DNA topoisomerase. The primase forms RNA primer, which is 10 bases, which serves to initiate synthesis of both the leading and a lagging strand. And the leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase enzyme 3, and the lagging strand is synthesized discontinuously in the 5' to 3' direction by the formation of Okazaki fragments. And DNA polymerase enzyme 1, it removes the primers, RNA primers, and replaces the existing gap with the appropriate deoxynucleotide. And DNA ligase seals finally the break of the Okazaki fragments as well as around the primers to form continuous strand. DNA repair in prokaryotic DNA replication is done by proofreading activity. Here, in bacteria, all the three DNA polymerase enzyme 1, 2, 3 have the ability to proofread using 3' to 5' exonuclease activity. When an incorrect base pair is recognized, DNA polymerase reverses its directions by one base pair of DNA and excised the mismatched base, and after the excision, the polymerase can reinsert the correct base and replication can continue. Next is the eukaryotic DNA replication. Here, replication is bidirectional and originate at a multiple origin of replication. DNA replication uses a semi-conservative methods. It occurs only in synthesis phase and at many chromosomal origins. Eukaryotic DNA replication takes place in cell nucleus. Synthesis occurs only in the 5' to 3' directions. An individual strand of DNA are manufactured in different directions, producing a leading and a lagging strand. And lagging strands are created by the production of small DNA fragments called Okazaki fragments that are eventually joined together. And lastly, the feature of eukaryotic DNA replication is that eukaryotic cell possesses 5 types of polymerase enzyme which is involved in the replication process. Next is the enzymes for eukaryotic DNA replication. These are helicases, single strand binding protein, DNA polymerase enzyme, and as I have earlier discussed in my previous slide about these enzymes functions, and the eukaryotic cells contain 5 different DNA polymerase. These are alpha, beta, gamma, delta, and epsilon. Alpha and delta, they are for the chromosomal DNA replication, and beta and epsilon is for the DNA repair, and gamma is for the mitochondrial DNA replication. DNA polymerase alpha and beta synthesize the lagging strand via Okazaki fragments. The RNA primers are synthesized by DNA polymerase alpha, which carries a primase subunit. DNA polymerase delta synthesizes the leading strand. And telomerase is an enzyme, a DNA polymerase enzyme that contains an integral RNA that acts as its own primer, which is used to replicate DNA at the end of the chromosomes, that are the telomeres. And DNA topoisomerase 1 function is that it relaxes the DNA helix during replication through creation of a nick in one of the DNA strand. And DNA topoisomerase second enzyme, it relieves the strain on the DNA helix during replication by forming in the helix through the creation of a nick in both strands of DNA. And the last one is the DNA ligase. It forms a 3' to 5' phosphodiester bond between adjacent fragments of DNA. Now, the steps in eukaryotic DNA replication. Here in this figure, it is showing the process of eukaryotic DNA replication. What is happening that replication of each linear DNA molecule in the chromosome start at many origins. On every 30 to 300 kilobase pair of DNA, depending upon the species and tissues, are proceed by the bidirectionally from each origin, and at each origins are replication bubbles form consisting of a two replication forks moving in opposite direction. The DNA replicated under the control of a single origin is called a replicon. DNA synthesis proceed until the replication bubbles merge together. As you can see in this figure, the bubbles are forming, and at the origins the enzymes unwind the double helix, making its components accessible for DNA replication. The helix is unwound by the helicase enzyme to form a pair of replication forks, and the unwound helix is stabilized by a single strand binding protein and DNA topoisomerases. The RNA primer then required, which is made by DNA polymerase alpha, which carries a primase subunit. And DNA polymerase alpha initiate the synthesis of lagging strand, making first the RNA primer and then extending it with a short region of DNA. DNA polymerase delta then synthesizes the rest of the Okazaki fragments, and the leading strand is synthesized by polymerase delta. The leading strand is synthesized continuously in 5' to 3' direction, while the lagging strand is synthesized discontinuously in the 5' to 3' direction through the formation of Okazaki fragments. And at the completion of synthesis, DNA ligase enzyme seals the break between the Okazaki fragment as well as around the primers to form a continuous strand. Now, the DNA proofreading activity in eukaryotic DNA replication. In eukaryotes, only the polymerase that deals with the elongation, that is delta and epsilon, they only have the proofreading ability, that is 3' to 5' exonuclease activity. It is known as proofreading ability. If an error is detected, the erroneous base is removed via 3' to 5' exonuclease activity and replaced with the correct base. And the excision repairs, it removes the pyrimidine dimers formed by the ultraviolet rays or other mutated base and replace them. Now, the significance of DNA replication. DNA replication is fundamental genetic process that is essential for cell growth and division. DNA replication involves the generation of a new molecule of a nucleic acid in which the DNA plays a crucial role for life. And DNA replication is important for properly regulating the growth and division of cells. It conserves the entire genome for the next generation. Thank you all.